Welcome everyone. I'm going to get started now. So today I'm going to be going into some really good detail on fire blight and focusing on the blossom blight phase. And I'm going to do that by going through the biology and then tying that into some modeling scenarios. Just some quick housekeeping. Because this is a webinar format, you all have your audio and video turned off during the webinar, so no need to be concerned about that. But if you do want to speak, I can give you that ability. So just let me know in the chat or the Q&A feature. I definitely encourage questions. So if you do have questions, please use that Q&A feature at the bottom of your screen. And I think we'll wait until the end of the presentation, just because that will be easier for me to be able to see what questions are there. So feel free to post your questions anytime and I'll address them at the end. All right, so the agenda for this session, first we're going to look at understanding the biology of fire blight, blossom blight. Then we're going to interpret modeling scenarios, and then overview of some blossom blight management options. And then I didn't want to ignore trauma blight. So even though we're focusing on blossom blight, I did want to throw in some notes about trauma blight as well. So like I said, the focus of this webinar is mostly on fire blight biology. So if you want to know more about how to actually use PomeBlight, I already did a webinar that acts as a video tutorial. So you can find that at farmdatatools.ca. We also have a quick start guide if you want to follow along. And what those resources are going to do is help you subscribe and register and set up for PomeBlight and review the key features about actually using that model. So we're going to focus more on the biology today. Now, before we dive into it, just an overview of fire blight disease. It is a bacterial disease of apple and pear trees, and infection is preventable, but of course, using antibiotics, it's very expensive. And also because of the label requirements, we have limited opportunities when those antibiotics can be applied. So the reason why we turn to modeling is so that we can identify those optimum management opportunities and hopefully save money and time by targeting those key periods. I would argue that we don't truly understand the biology of something until it can be reproduced in a model. And the Mary Blight model has been a really good resource for us. It's been, quite reliable, and there was years and years of research that went into the production of this model. So I've read the manual for Mary Blight cover to cover, and it explains, exactly how that model is functioning. And the PomeBlight model is based off of those Mary Blight calculations. So I want to be able to explain this model so that it makes sense, and that's what we're going to go through today. So starting off with the biology. So this bacterial beast, I just want to give you an overview of its entire life cycle before we zoom in onto that blossom blight stage. So the bacteria overwinters in cankers, and we've talked about that this year. We expect them to start oozing around the tight cluster to pink stage. That's when the bacteria become active, those bacteria that have stayed within the tree and stayed alive over the winter. And then that ooze, it contains bacteria, and the bacteria gets disseminated by the wind, particularly when there's rain as well, and from insect activity that's attracted to the sugars in that ooze as well. So it could be flies and also pollinators between flowers. So then that leads us to the blossom blight infection stage, and blossoms are a main point of entry because they have a natural opening at the base of the flower, which is the nectar pore. So that's what we're going to go into detail on. But essentially, if you have 1 to 4 of these cankers, for every 2.5 acres, that can cause enough ooze to create a blossom outbreak. So there's quite a bit of bacteria contained within those cankers and contained within ooze. So after a blossom infection, it can spread to the shoots, and the plant does have its own defense mechanisms, and that's why trees can form cankers to stop the spread of that bacteria. But the bacteria can also survive in those cankers, even though its movement is stopped. That's where it can continue to survive and live at the margins and then overwinter, and this cycle repeats. So we're going to focus on that blossom blight stage now. So in order for a blossom blight infection to happen, there's 4 key factors that the calculations consider in the Mary Blight model. And they're all based on biological reasoning that we're going to go into. So the first factor is that, of course, you need to have open blossoms. And we'll go into that in more detail. The second factor is you need to have bacterial reproduction so that there is enough bacteria present to cause an infection. And the third factor is the wetting, which we'll discuss is involved in bacterial movement. And the fourth factor is establishing those infections. So, movement of that bacteria and further reproduction when it's in the plant system. All right, so beginning with open blossoms. So the blossoms are the infection site for the bacteria during this blossom blight stage. So the bacteria, it's called Erwinia amylovora, and they survive and reproduce on the stigma of that flower. So here I've got the stigmas circled, and right at the top here, it's the perfect environment for that bacteria to grow. It's wet, it's nutrient rich. And this kind of bacterial growth is called epiphytic growth. So it's just living on plant tissues and growing and surviving. So bacterial populations build on these, and it's the wind and the rain splashing and the insect activity that is, distributing bacteria from cankers to this stigma site, or from one stigma that's already been inoculated with bacteria to another stigma that, can pass the bacteria along. So essentially, this is where the bacteria is growing, this little site on the flower. Now, just a little side note here about Nova Scotia. We live in a very humid climate, and there's this research article that shows this interesting information about how the relative humidity can impact bacterial growth on the stigma. So this image on the right-hand side of the slide begins with 60% relative humidity, and those green fluorescent dots are the bacteria. And so this is 2 days after bacteria have been inoculated on the stigma under these different relative humidity conditions. So you can see 60% relative humidity, there's some bacteria, 80% in the middle here, there's more bacteria, and 100% relative humidity, there's quite a bit of bacteria. So of course, those higher relative humidities do support more bacterial growth on the stigma. And fire blight infections then can happen more often and can be more severe under humid conditions like we tend to see in Nova Scotia. And that's attributed to them growing faster, like you can see there, but also they do tend to act more virulent in these humidity conditions. So they're actually more likely to suppress the plant host in humidity and induce plant cell death. So they're actually more aggressive under high humidity conditions. Okay, so the bacteria are growing on the floral stigma, and so the next step to talk about here is that bacterial reproduction. So I let you know that the bacterial growth is called epiphytic growth. So the Blight model uses this term epiphytic infection potential, which is abbreviated EIP. And that is an index that lets us know the infection risk because it's representing how much bacterial growth has happened. So it is a mathematical calculation here. And it's an index because it ends up being a percentage. So that bacterial growth keeps track of the accumulated heat sum, so the amount of heat that we've seen over a certain period of time, divided by the amount of heat that is required to have enough of a bacterial population to cause an infection, like an epidemic level of infection. So once the accumulated heat sum is equal to the threshold heat sum, that would be, you would reach 100%. So the EIP would be 100. And that would mean that you have the potential for an infection. So that's why we're always watching for that EIP of 100, because that's saying we've reached 100% of the threshold. And that EIP isn't calculated indefinitely. It's calculated during a floral lifespan. So there's a heat sum that a flower stays alive on which the bacteria are growing. So, so that's kind of how that formula is continuously calculated each new day. So basically, the bacteria love heat, and so their growth is favored if temperatures are above 18.3 degrees Celsius. And like I said, that growth is calculated over a floral lifespan. And so when you're looking at the model, it's considering the infection risk based on the oldest open flower that is able to support bacterial growth. So usually flowers last about 3 to 4 days, depending on the temperature. So your model is telling you what your 4-day-old flower risk would be. So just to keep it simple though, your EIP increases during warm weather, your EIP decreases during cool weather. So again, that's a representation of your bacterial growth. And that bacterial growth on the stigma is not directly harmful to the plant, but it increases the potential for infection. So that's why we're monitoring for how much bacteria is present. So I mentioned that there's a time frame for bacterial reproduction, and that's because the flower ages over time and the stigma becomes a worse host of that bacteria over time. So this is an awesome photo to show that concept. So A and B here are showing a newly opened flower. So this is the stigma of that flower. It's really healthy. These papillae are, nice and turgid and supportive of bacterial growth. But then figures C and D here are a 4-day-old stigma on a flower. That flower is quite old at this point. It's covered in mucilage and bacteria on the close-up here. But, essentially it's going to run out of that wet and nutrient-rich environment, and the bacteria are no longer going to colonize and grow on that stigmatic surface. So we've got these flowers aging over time, and that's why the model is only tracking the bacteria on flowers up to basically 4 days old. So why does this really matter when we think about different varieties? Some varieties are at higher risk because they have an extended bloom period. So what happens is on a single tree, you've got flowers with different ages simultaneously. So if you've got an old flower that has this population of bacteria that's been able to grow, and then you've got a newly opened flower right beside it, then there's the potential that bacterial population can very quickly move on to new flowers. So what happens is it lengthens the risk period for a particular variety if it has that extended bloom period, and there's a longer total time for that bacterial growth on an individual tree. And then also it becomes even more risky as the bloom season progresses, because you've had so many flowers that have the potential for bacterial growth on the stigma, that when you've got old flowers appearing, there's so much bacteria broadly dispersed in the landscape that it can very quickly be distributed to new flowers that are blooming late. So it gets even more risky over time. So this question comes up every year: when are flowers no longer at risk? So like I said, the stigmas no longer support bacterial growth when they get too old, but you're not out there in the orchard monitoring when a flower opens so that you can say it's been 3 to 4 days. So essentially the only way we can practically say that a flower is no longer at risk of infection is when it's at the petal fall stage. So protection is needed until petal fall. And like I said, you don't want to underestimate those late blossoms, because they have had ample opportunity for becoming inoculated, with that bacteria being broadly distributed in the landscape. Okay, so at this point, we've talked about having open flowers to have that environment where the bacteria grows on the stigma. We've talked about bacterial reproduction happening in the heat. And then the third factor here is that a wetting event is required for the movement of that bacteria. And it allows the movement towards the nectar pores, which is the natural opening in the flower where that bacteria is going to gain entry to the plant system. So again, a really neat photo here that's been shared in research. And what this photo is showing is it's a close-up of the stigma again. And here you can see that there's this groove that moves from the stigma towards the base of the flower. And part B here just shows that groove in closer detail. And part C is really, really close, where we're just seeing the groove along the stigma. And so this characteristic of the stigma actually encourages the movement of the bacteria. If there's water, it encourages the movement of the bacteria from that stigma down towards the base of the flower. So, wetting, which could be a rainfall or a heavy dew, create that channel of water to allow that bacteria to move. And so that's why we often see blossom blight happening in low areas of the orchard, because dew tends to be quite common in those situations. And dew can create a very consistent channel of water within that groove to allow the bacteria to move. Okay, so we've got the flower as the first factor. We've had bacterial reproduction, then we had wetting for the bacteria to move, and now we're at the point where infections become established. So now we're at the base of the flower, where there are these nectar pores where the bacteria can gain entry. So we're actually looking at a close-up photo here of a nectar pore that's circled in yellow here. And in blue is showing the bacteria that are very near to that nectar pore. So there's bacteria everywhere along the base of the flower here. And this is a 3-day-old flower, so you're seeing some of that bacteria that has already reached the base of the flower. And then in order for these infections to become established, we do need some warmth for the bacteria to be able to move and continue to multiply and enter the nectar pore. So that would be a daily temperature of 15.6 degrees Celsius. That's what's used in the model. And it influences that rate of migration to the nectar pores, and also once the bacteria enter the plant system. So once the bacteria enter the plant through a blossom infection, then they can follow the vasculature of the plant. So that's why we tend to see the symptoms showing up along the midrib and the veins of the plant. And so you see that those brown streaks, and the infection progresses down to the shoot as well. And you can also see browning if you scrape below the surface there. And so it seems that the bacteria follow the vasculature. They're probably in the intercellular spaces along the vein, which isn't really detail that you need to know, but it's interesting because apparently if the bacteria actually go into the xylem, they can starve in there. So they're probably right next to the xylem and not actually within it. So interesting fact. But yeah, basically you can see the browning, and they do travel along the vasculature, meaning that they can go throughout the plant system very quickly. So just to summarize now, a blossom blight infection, again, can be produced when those 4 factors are occurring. So the model calculations are based on those 4 factors. And the Mary Blight model presents the risk as low, moderate, high, or infection, and it's based on the number of factors that are present or absent. So if you don't have blossoms, of course, there's no risk of a blossom infection, but as soon as you do have a blossom, then it's considered to be a low risk. And then if you have a blossom and another one of those remaining factors, then it's moderate, and so on until all 4 factors produce an infection. So just a quick summary here, bacteria on the stigma, on an open flower, reproduction, that's then washing the bacteria down into the base of the flower, into the pore. Okay, so that is the main biology I wanted to cover up front there before we start looking at some of these scenarios. And I'm going to be using some screenshots from the PomeBlight model. You can access that from farmdatatools.ca. You don't have to be subscribed to the PomeBlight model to be able to benefit from the following scenarios. There is still the Mary Blight model available for free, but you have to enter everything manually. So the main benefit of the PomeBlight model is that we've created an automatic connection to the weather stations and a much more friendly user experience. So if that's what you're interested in, then that is an option now. So as we dive into the model, I don't want you to forget the biology that plays into it. So the model is showing you those exact 4 factors that we talked about. So when you're looking at the model, we've got whether or not blossoms are open in the phenology section. We've got whether or not there was a wetting event, which would be a rainfall or a dew. And so there's a dew in this case, and that's highlighted in red because that's one of the factors being met. The average temperature, which would have to be warmer than 15.6 degrees Celsius, would also be highlighted in red. And then the epiphytic infection potential, which we discussed, represents bacterial growth. And if that's greater than 100, which would be 100% of the bacteria you need to cause an infection, then that would also be highlighted in red because that's another one of the factors being met. So in this case, all 4 factors are met. So the infection risk is an infection prediction. So the first scenario we're going to go through is a simple one, and it's deciding when to spray, but in a simple kind of scenario condition here. So the scenario is, you're asking yourself, is an infection coming? When should I spray? And how long will flowers be protected? So these are the common questions, and we're going to look at some common conditions. And this is everything you should be able to answer for yourself when you're reading the model. So here's the scenario that I've put together, and I've entered in some minimum and maximum temperatures to produce some bacterial growth, basically. So in this example, and it's not occurring right now, this is just an example, but in this case, infection is being predicted on the next day. So the first line is the current day, and the next line is the next day. So an infection is being predicted, and how we can tell is by looking at the bacterial growth under the EIP, and the infection risk is saying infection. So what you would do in this case is, you would look at the current date. There's quite a bit of bacterial growth there already, so you might wonder if applying an antibiotic spray on the current date would prevent the infection for the following day, and that way you could get that antibiotic on before the upcoming rainfall. So what I'm going to do is input an antibiotic spray on the current date. So I've put in that antibiotic spray, and after that you can see it has completely knocked back the bacterial population represented by the EIP. So now that's 0 and that's reset. And so the next day isn't going to reach that threshold bacterial level because it's been wiped out and it's starting back over again. So in this case, that infection has been downgraded from infection, and then it goes down to a high risk still because you still have all 3 of the factors. You just don't have enough of a bacterial population to get washed into the base of the flower. So to answer that question, is an infection coming? Yes. When should I spray? It works if you sprayed on the day before the infection. And how long will flowers be protected? Well, according to these temperatures, it looks like an infection won't happen again within the next 3 to 4 days there. So you can continue to monitor that situation. Let's see. So I think I had already addressed this. I just want to make it really clear that the EIP is an important factor to follow because it does represent the bacterial population. So even though it's still saying the risk is high, you do need to check and see how much bacteria is present. And in that case, putting the antibiotic spray on does avert the infection risk. So just to show that in a picture here, of course, you have bacterial growth and there's water. So of course, it's going to result in an infection. But if you put the antibiotic on, you don't have the bacterial growth. So there's no risk of infection as long as you've had good coverage of your antibiotic on the floral stigma. Now, just a side note here as well, what do you do in the model if you need to spray from 11 p.m. one day to 2 a.m. the next day? So how would you input that spray into the model? So the thing is, the model is a daily model, and so it's not sensitive enough to track on an hourly basis. So we can't see that hourly bacterial growth. Which the developers say is not practical anyway, because you're not going to be able to spray on an hourly basis anyway. It takes, so long to get through an orchard. So the daily prediction is kind of the most practical anyway. So what you would end up doing in this situation is a nighttime spray is treating the flowers that opened on that day. So you're going to choose that earlier day for a conservative approach. Because what you're doing is you're protecting the flowers that opened earlier and not the flowers that are about to open on the current day after 2 a.m. So the next scenario here is the case of explosive bacterial growth, and this can happen sometimes. what happens is on a single day, it's hot enough that if a flower does become inoculated with bacteria, that bacteria can grow fast enough to reach such a high level that, that it could cause an infection. So in this scenario, we're thinking about these new flowers. So this is a video here that I'm going to play. And so the flowers that are already opened, say you put on an antibiotic spray and the bacteria has been knocked back on these flowers, knocked back to 0. But when I press play here, after the spray, these flowers are going to open. And if they become colonized by bacteria, that's where the population can build within a single day. So just remember that the model tracks those 4-day-old flowers, but in the case of a really hot day, what you can do is if you apply an antibiotic in the model, which you're going to see on the next slide, it's going to reset the population to 0 on those really old flowers, but it's still going to tell you the bacterial growth on newly opened flowers. So we're going to look at that in a second. Okay, so in this case, we've got an example where there's no antibiotic spray and it's going to be really hot on the next day. So it's going to get up to 30 degrees. And you can see that the prior day, there was only a level of 36 representing bacteria. And then that EIP value goes up to 145 the next day. And then what you can do is, like, that 36 is representing the older flowers, and the 145 is still representing the older flowers. But if you enter an antibiotic spray, all of those older flowers that got treated with the antibiotic spray are set to 0. And you can see that the EIP, the bacterial growth that follows on the next day, is still reaching 109. Because that's representing the new flowers that opened and how much bacteria is actually just growing in that single day. So your old flowers are protected, but your new flowers are actually susceptible to infection in this case. So that's where you would kind of play around with this antibiotic spray, and you would see, like, there's no point in putting it on if you're just knocking back a small bacterial population on the day before. And you would really want to try to target those new flowers, if possible, on the day that the bacteria is really going to grow. So you wouldn't put the spray on that day. You would try putting it on the day with that bacterial growth. Okay. So going into the next scenario here, would be considering how to choose your bloom date. So the question is, how do I pick the first bloom date, and should I monitor varieties separately if they have different first bloom dates? So luckily, the answer to this is that we don't need to overcomplicate it. We don't need to monitor these varieties separately. Especially if we are 4 days into bloom. And the reason for that is that the model, like I said, represents bacterial growth on flowers up to 4 days old. So if you have continuous bloom, then those varieties are going to become synchronized, and all of those newly opened flowers have equivalent risk of bacterial growth. So what you want to do, though, is you do want to pick the first day in your orchard that a blossom opens, because you do want your model to be able to reflect the oldest open flower and its specific infection risk. So you are picking the first day that blossoms open in the orchard. Yeah, but as time goes on, like I said, those varieties will become synchronized. So you do want to get protection on all varieties that have open flowers at the time of an infection. And of course, if there are no flowers open, then there's no risk of infection. And I also wanted to mention here that in the model, you do have to enter a green tip date, but it doesn't actually matter for your blossom blight predictions. It's just using that green tip date for canker blight predictions. So you don't have to be, if you're not concerned about canker blight predictions, you're only concerned about blossom blight, then you don't have to be like 100% accurate with your green tip date, but you do still need to enter one. So I just want to show this, this scenario in the model format. So because a bacterial growth happens within the first 4 days, roughly, of those flowers opening, what you'll see in the model is that say on the first day, bacteria has built to 109, and then for the following days, the temperatures mean that that bacteria isn't going to, it's not going to change. It's just going to remain constant. It's not going to grow or decrease. Then it's just saying on the, so, you know, you just started the model on those 1 day old flowers, it's going to be 109. Then they're 2 days old, it's going to be 109. They're 3 days old, it's going to be 109. They're 4 days old, it's going to be 109. When they're 5 days old, they're not supporting bacterial growth anymore, because remember that stigma has aged. So it's the model is then going to say it's 0, because those new flowers that are opening under these temperature conditions, the bacterial population is not building. So I just wanted to show you here, that's how you see that 4 day, how it's tracking those 4 day old flowers. And then it shifts towards newer flowers as time goes on. Yeah, so that's how it becomes synchronized, because once you've passed 4 days, all of the flowers are the same age, and the model will be telling you the bacterial population on all of those new flowers. Now, the fourth scenario here is related to wetting events. So the scenario is the EIP is near or above that threshold level of 100, and the risk is high, but the wetting is the only factor that has not been met. So what do we need to consider in this kind of situation? So in this situation, if the EIP is near or above the threshold of 100, that means there is enough bacteria on the stigma, and the bacteria only needs to move down the groove to get into the base of the flower. And it moves by using water. So water can be a heavy dew, which needs to be entered into the model by the user, because the model can't predict that automatically. It can be 0.25 millimeters of rain, or it can be a high water volume sprayed by the sprayer, unintentionally wetting the flowers. So you want to be really careful when wetting is the missing factor, because any of these wetting events can lead to infection. So just showing that in the model here, what happens is you have enough bacteria. So looking at the EIP, it's 121, so it's above 100, and the infection risk is high, because we don't currently have a rainfall or a dew put into the model here. But then if you do have a rain, that rain is shown in red here. I put in 0.25 millimeters because that's the minimum amount to cause an infection. And you can see that infection has been triggered. And then if it was instead a dew, you would enter yes to a dew event, and again, an infection is triggered. So you really do want to watch that bacterial population and to be aware of which factors are missing, so that you can you can decide how safe it is to, trust that those factors are maybe not going to happen, or maybe they are going to happen. So just a little side note here about dew. Dew is actually very important because it creates a channel of water on a really high proportion of flowers in the orchard, more consistent than like a light rain would. So there's your dew on the groove of the stigma, and it ends up being perfect for bacteria to move. So definitely don't ignore those dew events. And like I said, low areas of the orchard tend to be more prone to fire blight infections because they do tend to experience dew more frequently than the rest of the orchard. So the next scenario that you might see in a modeling program is the carryover wetting. So in this case, it isn't raining today, so why is the wetness requirement already satisfied? So, you can see in this picture of a flower that the flower can hold moisture for a long period of time. It can hold it in the petals, it can hold it, in the groove of, coming down from the stigma. So what happens is if you have a rain more than 2.5 millimeters one day, it will allow that water to stay in the base of the flower, and it will allow bacterial movement the next day. So that's why you'll see in the model, if you have a wetting event more than 2.5 millimeters, in the next day, we've got this asterisk in the PomeBlight model, where you can hover over it and it's telling you that there's still water present there, even though there's zero rainfall on the current day. So that can still act as your wetting event because that water is still sitting there. And then a side note about water, there's sometimes this misconception that water washes away bacteria, and this is not true. Bacteria use that water to survive and disperse and colonize new flowers, like we've already talked about. So the bacteria loves the water, and there's billions of cells, so even if some of it gets splashed to another tree, there's still a lot of bacteria that would remain where it is. So I think this misconception came from rain events often corresponding with cooler temperatures. So it's actually the cooler temperatures that are responsible for less bacterial growth rather than the water or the wetting event itself. So just to show that in the model here, if you have rain and a constant temperature, so it's a minimum of 13 degrees and a maximum of 19 every single day, and say you do have a rainfall on that second day, you see that the bacterial population is not at all affected by the rain. So it's still there the whole time. But if you have a rain and a low temperature, so a low of 13, a high of 19, but then when you've got that rain event, you've got a low of 5 and a high of 10, then that's when you see the bacterial population decreasing because it's related to the cool temperature. It's not related to the wetting or the rain event. Now, the next scenario here is determining your degree of safety. So let's say an infection is not currently predicted, but you want to know like what happens if the forecast changes, because we often see that the temperature is several degrees warmer than it is forecast. So you want to be able to play with the model to see, how comfortable you are in case that forecast changes. So what you would do is you could edit the temperature, and let's say So I've got a high of 22 degrees here, and the corresponding bacterial population is 85. So what happens if I increase this to 23 degrees? And you can see the bacterial population increases to 97. So 97% is getting very concerning. Oftentimes, you know, we find locally that 97 is enough to cause an infection too. So you wouldn't necessarily want to wait exactly for that 100 threshold. But then what happens if it's actually 24 degrees? Well, then you're easily over 100 in this case, and an infection would be predicted. So then you would ask yourself, how comfortable am I if the forecast is 22 degrees, but what if it actually gets warmer? So when you're getting close to 100 EIP, that's when you have to start making some decisions, and the model can help you determine your degree of safety. So you can definitely play around with the forecast, especially in that 1 to 3 day prediction period. Now, the next scenario here is with regard to micromanaging. And so the recommendation is don't micromanage. What you need to do is read the risk. So say the EIP is above 100 today, but it won't rain until tomorrow at 1 a.m. That's getting too much into micromanaging, because you would be trying to interpret this model hourly when it's actually a daily model. So in that case, like, technically an infection is not predicted, but you know, when we look at the model here, technically an infection is not predicted because you've got the high bacterial population on one day, but it's not going to rain until the next day at 1 a.m. But you know, you have to think about the model as a daily snapshot, but bacterial growth is a continuum. So just because one day ends doesn't mean that bacteria is not going to be there if there's a rain event an hour later. So I think generally our industry is pretty good at watching that EIP value. And if it gets up to 133 and there's rain in the forecast, you're not going to wait and see when that rain, you know, exactly happens. So, you know, just keep that in mind when you're looking at the model. You do have to interpret it. You know, if you see that you have a high risk, you want to find out when your next wetting event is going to happen. And if there's a risk that it could wash that high bacterial population into the flower, then you want to make sure you're covered up for that kind of a risk. All right, so those are all the scenarios I'm going to go through today. And now I want to talk about blossom blight management options. So you understand the biology, and you've got access to the model to be able to play around with some different conditions, and that can influence your management decisions here. So first of all, you know, one approach is to pinch the buds off. And the reason this works is because you're removing the stigma and you're removing the nectar pores. So the bacteria can't grow on the stigma or be washed into the nectar pores. So it does work. It's just very time consuming. So it can be an option for young non-bearing trees if that's an approach you want to take. But the issue is there's a very narrow time frame when this can be done. So usually you would want to start around the bud separation stage, because that way you can avoid pinching off the terminal shoot. So you can at least see where the terminal shoot is. But then, you know, you really want to do this pinching before bloom time, because if you were to pinch around bloom time, you're touching the stigma where the bacteria is growing, and you could be transmitting that bacteria as you're wounding the trees going down the tree row, pinching flowers. So you definitely don't want to do any pinching of these tissues when it's wet and dewy. And if you're doing this during bloom time, you don't want to do it when the EIP is high, because there would be high bacterial populations on the stigma. What you would want to do also is leave 1 to 2 days of warm and dry conditions for those wounds to heal, because that way the bacteria don't have a wound to be transferred into. So it's a very time-consuming process with a very narrow time frame, not very practical. And if you miss some of the blossoms on these trees, then you may want to spray anyway during an infection risk. So, you know, you have to decide if this practice makes sense. Another management option is apogee or kudos, which are perhexadione calcium as the active ingredient. And they can help to reduce shoot blight incidence and severity, which can then prevent the initiation of cankers if you're able to cut those shoots off quickly enough before that bacteria progresses further into the tree. So if you're reducing the amount of cankers, then you're reducing your inoculum for bloom the following year. So how this product works is it increases the thickness of the plant cell walls. So you know how the bacteria is moving through those intercellular spaces and along the vascular tissue. Well, you're increasing the thickness of the cells that it's moving through and around, and that creates some structural resistance to slow that progression down. The product is not systemic though, so make sure you get good coverage of all of the plant tissues. And if you're looking at putting it on young trees, usually a lower rate is used. So 1 to 2 applications at 300 grams per 1,000 liters of water. And you would expect to see some growth reduction from that treatment, but the priority is really in preventing tree death, especially on those very susceptible varieties. And the label also says to do that application 14 days later. So you're, you know, making sure that you've got that continuous structural resistance. Okay. So now getting into management products during blossom blight, or I guess I should say first beforehand, we do have those copper-based products that can be applied at or just before green tip, which, you know, we've already passed now for fresh market varieties. But basically what the copper does is it's not killing bacteria inside the cankers. That copper is just coating the plant surfaces so that when the bacteria might be transferred to those plant surfaces, it's not able to grow and colonize like the bark and the bud surfaces. So you're really just reducing the amount of bacteria that's present in the orchard before bloom time to hopefully reduce the risk during bloom. But then during bloom, we've just got the blossom sprays, which are antibiotics. So we've got streptomycin or cassumin, which are registered on both apples and pears. And what these antibiotics do is they kill and inhibit the bacterial multiplication on the stigmatic surface. And if it makes sense, it's best used the day before an infection event, unless you've got that explosive bacterial growth that we talked about that can happen on a single day. But it's best if you can knock back those bacterial populations before a wetting event lets them travel into the flower. There are some differences to note about streptomycin and cassumin though. And cassumin tends to be much more finicky. So streptomycin is more flexible because it's partially systemic. So that streptomycin can actually chase after the bacteria to a degree. So once the bacteria goes into the nectar pores, the streptomycin can as well. And as long as it hasn't been long enough for that bacteria to really start to multiply, then the streptomycin can knock back that population enough to prevent infection. But in the case of Cassumin, we rely on that only to knock back the bacterial population on the stigma. So it does not chase after the bacteria. So you would not want to put on Cassumin if the wetting event has already happened and allowed that bacteria to get into the nectar pores. So, for example, it makes me really nervous if we're in a situation where we've got that carryover wetting event. So the wetting happened the previous day, the flowers are still wet. I would be concerned about putting Cassumin on just in case the bacteria have already entered the nectar pores. Cassumin is also UV sensitive, so it's recommended that it's sprayed at night so that that product is still a high concentration when it's sitting on the stigma and knocking back the bacterial population. But I mean, it's still a handy product to have because it can be applied 4 times per year during bloom, and it has a good reentry interval of 12 hours. So I just wanted to bring up those concerns though, so that you're aware that Cassumin is not as flexible, but it can be great for working in young trees with that short reentry interval. Okay, the other difference between strep and Cassumin is related to their rainfast period. So if streptomycin dries, then it's likely to absorb and have good activity. But in the case of Cassumin, if it doesn't have the chance to absorb, then, Cassumin does not absorb because it's not partially systemic. So you want to make sure that Cassumin has a couple of hours to sit on the stigma so that it's knocking back the bacteria. So you don't want your Cassumin to get washed off, whereas you might have a little more flexibility with streptomycin with its ability to be absorbed. So Cassumin definitely more risky in a downpour situation where it could get washed off, but a light rain might be okay, but still be careful about the Cassumin. Okay, I'm getting short on time here, so I think the industry is aware of prioritizing, susceptible varieties. Don't ignore Honeycrisp, especially last year that got some people into a difficult situation. The bacteria can certainly still infect Honeycrisp. I do want to also say spraying every second row is also risky because you have to think about you're trying to get that antibiotic onto the stigma and the flowers are really puffy. So, you want to make sure that you're getting good coverage on every single row. And waiting for post-infection is also risky because you've only got like up to 20 to 24 hours before that bacterial growth becomes explosive within the tree system. So when you're using the streptomycin to chase after it, you've got a very limited amount of time. Interesting note here from a fire blight school that I attended this past winter. It's from the US and it was a national collaboration. Somebody did a really in-depth presentation on biological products, which are your alternative products, and concluded that they will not avoid disaster. So they are not good killing agents. So if you already have a bacterial population on the stigma, they're not good at killing it. Where they might be helpful is in working as protective agents. So that becomes a lot more complicated with, continually applying those products and being aware of what kind of bacterial populations are present. And we definitely don't have experience with that locally. And another point here that I learned is that the bacteria overwinters better in cankers of your resistant hosts because they're resistant and they're going to stay alive and they're trying to fight the bacteria, but the bacteria just kind of gets, secured in a canker and is able to become active every year, whereas a susceptible host is likely to die and take the bacteria with it because the tree dies. So having infected resistant hosts can increase your inoculum level in the orchard. There is the opportunity to test for fire blight. So we do carry some of these tests at Perennia if you're interested. And it's kind of like diagnosing yourself for COVID. So positives are certain, but you can get false negatives if you've sampled the wrong place in the tissue where the bacteria are not present. So you can contact me if this is something you're ever interested in, if you think you have fire blight, and I'll be taking the live tissue on the leading edge where we would be able to detect it using this kind of procedure. But we are charging a small fee for these kits just to cover the cost of the samples. I think it's around $15. So, just to cover the cost of those supplies. So I'm running out of time here, but just a few notes about trauma blight. We all know that opening these kind of wounds allows the bacteria to get in, and the cell death there actually adds nutrients to let the bacteria grow. But I just want it to be clear that in the case of trauma events, the bacteria are already present on the plant tissues and can be easily transported from oozing infections. So you don't need the heat like you do for the bacteria to grow on a stigma, because in this case, the bacteria are already present and just need to be transported to the wound in order to cause infection. And why is it important to work in dry trees? Because, we've already talked so much about how important water is to the bacterial movement. But in the case of rain, it actually rehydrates dried ooze. It splash disperses that bacteria. Bacteria becomes airborne near active infections, and the bacteria is present in very high populations in the droplets on infected trees. And then if you are working in those trees and you create a wound, well then, that water that's carrying bacteria can easily enter that wound, and the plant doesn't have defense mechanisms against that kind of entry. The chance of a wound becoming infected diminishes 48 hours after injury. So that's why if you are going to be working in trees causing wounding, then if you can allow 2 days for that to dry off and heal, then that can reduce your chances of infection.